nucleic acid for DNA and RNA. The structures, for simplicity, are divided into two parts. We can say primary structure of nucleic acid and secondary structure of nucleic acid. Let's discuss first primary structure of nucleic acid. Ribonucleic acid and deoxyribonucleic acid, that is DNA and RNA, are polynucleotides, or you can say types of nucleic acid. Hydrolysis of these nucleic acids with alkali or enzyme gives corresponding sugar nucleotides. RNA will give ribose sugar, whereas DNA will give 2-deoxy-D-ribose sugar. Alexander Todd suggested that the nucleic acid molecule has a backbone chain which consists of alternating sugar and phosphate unit, whereas the bases are attached to sugar as a side chain. It can be represented in a simple manner like this. Here we can see a basic backbone or a polynucleotide chain consists of sugar and phosphate alternatively arranged, whereas the bases, purines or pyrimidine, they are attached to the sugar molecule, okay, as a side chain. This primary structure here particularly represent a two-dimensional structure. When we talk about these nucleotides, they are joined together by forming a phosphate ester bond. That bond is formed between 5-hydroxyl group of one nucleotide and 3-hydroxyl group of the sugar on the another nucleotide. One end of the nucleic acid polymer has a free hydroxyl group at the C3- carbon, whereas the other end has phosphate at C5- carbon. Hydrogen ion titration of these purified RNA shows absence of second acidic proton. Thus, it proves the presence of phosphodiester bonds in the nucleotides or polynucleotide chains. Now, these can be summarized in a following manner. Here, we have a backbone chain for DNA molecule which consists of this 2-deoxy-D-ribose sugar and phosphate unit as a backbone, whereas the bases are attached to the sugar at C1- carbon. Here we can see for this sugar molecule, this 3-OH hydroxyl group is attached to phosphate, which is attached to 5-OH of another sugar. So this is how the phosphate esters are formed, or these are also called as phosphodiester linkages present between nucleotides. Secondary structure, particularly we are going to discuss for the deoxyribonucleic acid. So Wilkinson and his X-ray studies shown that DNA molecule have a double helical form, also known as double helix structure, which was proposed by Watson and Crick model. This model says DNA consists of two polynucleotide strands which are coiled in a right-handed manner around each other, and they runs in opposite direction. I am repeating. Watson and Crick model suggests that the DNA consists of two polynucleotide strands which are coiled in right-handed manner around each other, and they run in opposite directions. The C3- to C5- in one strand, while C5 dash to C3 dash in the other strand. These are the directions. Okay, when we talk about the two strands, they run in opposite direction. For one strand, it is C3 dash to C5 dash, and for the other, it will be then C5 dash to C3 dash. How it affects? It affects the proximity of purines and pyrimidines. Because of these opposite directions of these two strands, purine and pyrimidine bases in these two strands face towards each other for effective hydrogen bonding. So now let's see a double helix structure for the DNA. Here we have two strands represented in this yellow strip, which are consist of sugar and phosphate as a backbone, okay, whereas the bases which are attached to the sugar as a side chain. This particular helix has two grooves, major groove and minor groove. One complete helix has a distance of 34 angstrom, which suggests that 3.4 unit per turn of this double helix structure. These purines and pyrimidine bases, which are facing towards each other because of the anti-parallel nature of these polynucleotide chain, forms hydrogen bond between them, and because of these hydrogen bonding, these two polynucleotide chain are held together. Now, when we talk about the bases, purines and pyrimidines, which are present on the two nucleotide chain, okay, in the double helix structure of DNA, the pairing is according to the Chargaff rule, suggests that the percentage of guanine plus percentage of adenine divided by percentage of cytosine plus percentage of thymine is nearly equal to 1. The two rules given by Chargaff, okay, are helpful to understand this base pairing. The second rule says the percentage of adenine to the percentage of thymine and the ratio of percentage of guanine and percentage of cytosine are equal to 1. It means that the two chains which are held together by hydrogen bond between specific pairs of bases, the number of adenine are equal to number of thymine in the DNA, whereas number of guanine are equal to number of cytosine in DNA or RNA. The double bonds or hydrogen bonds present between adenine and thymine are 2, whereas 3 hydrogen bonds are present between guanine and cytosine. It can be represented with a structure in the following manner: adenine and thymine having 2 hydrogen bonds between them, and guanine and cytosine has 3 hydrogen bond between them. This is the structure of thymine adenine, showing the 2 hydrogen bonds, whereas cytosine and guanine having these 3 hydrogen bonds. And this represents the pairing of bases in the nucleic acid.